Good day, my name is Ellaina Wyllis. I am a sophomore and biology major at the University of the Virgin Islands. This past summer, I've been working alongside Dr. Lavita Rogers, who is an assistant professor at UVI, and our research focuses on an analysis of gene expression variability in asthma. So asthma is an inflammatory disease that targets the airways of the lungs. So unlike a normal individual's airway, an asthmatic airway tends to be much narrower and inflamed. And this is especially true in the presence of allergens, such as pollen and dust. Now what these allergens tend to do is that they exacerbate this condition, and that can sometimes lead to an asthma attack. And that's where the individual presents with coughing, wheezing, chest tightness, and shortness of breath. The Centers for Disease Control and Prevention reported that over 339 million people worldwide currently suffer from asthma. It is also one of the most common chronic diseases affecting children. Now, apart from its high prevalence, we also selected asthma as our disease of study because of how complex it is. So asthma is actually considered to be a multifactorial disease, and what that means is that there are many factors influencing a person's susceptibility to developing asthma. That can include gene-to-gene interactions, as well as a person's environment. Now, for our research, our aim was to identify human genes that may be playing a role in the onset of asthma. In order to do so, we needed to use microarray data, preferably from a large enough cohort that consists of both asthmatics and non-asthmatics. Now, microarrays usually come in the form of these small rectangular slides, on which RNA or mRNA is then used to detect the expression of thousands of genes. And we're able to do this because of the role that RNA plays in our central dogma. It is the means by which our genetic information is able to be translated into proteins. Now, because of this, we're able to use microarray technology to scan for which genes are being expressed and which aren't. Now, our initial hypothesis was that both sex and disease states are factors that are affecting gene expression in individuals with asthma. And we shaped our research question this way because many studies have found asthma to be more prevalent in younger males and older females. So this is the pipeline that we used for our project. It was previously formulated and published by Dr. Lavita Rogers and Dr. George Myers. And our first step was to conduct our data curation on Gene Expression Omnibus, or GEO. And that's where we selected for the data sets that we wanted to use, or the microarray data that we wanted to use for our analyses. So then we downloaded and pre-processed that data in R. And then we conducted our analysis of variants, and that's where we were able to identify the differentially expressed genes. And then we conducted a few gene and pathway analyses, which helped us get a better understanding of the functions and cellular components that are associated with these differentially expressed genes. So after conducting our data curation, we selected for these 4 data sets from GEO, which made up our cohort of about 800 individuals. And these are graphs depicting the demographics of our sample with regards to disease state and sex. So after conducting our analysis of variants, it identified 1,641 differentially expressed genes based on disease state. And we also conducted analysis for those influenced by sex, and we were able to identify 9,000 differentially expressed genes. However, because the majority of these genes were associated with sex-related differences like hormones, we later concluded that there are no interactions between disease state and sex. So this is a heat map of the top 10 differentially expressed genes based on disease state. And these are 2 of the most overly differentially expressed. So you can see that from the darkest shade on the heat map. Now, the CLC gene, or for these genes, what we did was we looked at the associations so that we could get a better idea of how they might be influencing the development of asthma. So the CLC gene in particular has been associated with allergic rhinitis, which is a condition that has been characterized by inflammation of the nose, as well as other symptoms like the tearing of the eyes in response to an allergen. So the CLC gene has also been associated with hypereosinophiliac syndrome, and that's when you have an infiltration or elevation of eosinophil in the bloodstream. Now, eosinophils, they are a type of white blood cell that, or whose role in the immune system is to cause the inflammation and the dysfunction of the cells that they target. Now, between these two associations, allergic rhinitis and hypereosinophiliac syndrome, we're able to make connections and inferences as to how this gene might be influencing the onset of asthma. And we're able to do this because we know that asthma is an inflammatory condition as well. Now, the RNA-SE2 gene has mainly been associated with pulmonary eosinophilia, and that's just an infiltration of eosinophil in the lungs. And what this tends to do is that it constricts the airways. And we know that that's also another phenotype that has been associated with asthma. So we also conducted a Reactome analysis, which is a tool that helps us identify the processes occurring within the cells, at least within the cells containing our differentially expressed genes. So these are 4 enriched pathways that our analysis identified. And we found that neutrophil degranulation had the highest gene count and was the most statistically significant pathway. So neutrophils are another type of white blood cell that contain these granules. And so what happens is that in the presence of an invader, such as certain bacteria, these neutrophils will degranulate or release the contents of their granules, which usually consist of antimicrobial and pro-inflammatory proteins. So once again, we're seeing that connection with this pathway to asthma with regards to an inflammatory response. We also conducted a Kegg analysis, which identified these top 4 gene ontology terms. Hematopoietic cell lineage it starts off with these stem cells, then give rise to a multitude of other blood cell types, including that of the neutrophils and the eosinophils that I discussed earlier, as having a huge role in our immune and inflammatory response. Our analysis also identified glioma and prostate cancer, which some studies have found to be associated with asthma, or at least a history of asthma. So the broader goal of our research is to use these gene and pathway associations towards improving asthma diagnostics, treatment, and preventative care. Now, this includes understanding the pathogenesis of asthma, as well as being able to design genetic testing that could even be used towards determining a child's risk of developing asthma. We could also use our findings towards gene therapies, where we'll be able to manipulate someone's genetic information or gene expression in an attempt to cure or treat them. So some of our next steps include a post-hoc analysis, which is where we're able to determine which of the differentially expressed genes are being upregulated and which are being downregulated. And then we'll be continuing our data curation as well, mainly for the purposes of conducting an age-specific analysis, where we'll be able to determine whether age is a factor that is influencing gene expression in asthma. So I'd like to acknowledge the UVRI's program by means of the NIH for funding this research, as well as Dr. Lavita Rogers for her mentorship and assistance on this project.